Hi, folks. Today we are nearing the end of our journey through the different types of biomolecules. We're going to start again by sorting out some of your molecule cards. The ones you have left are all lipids and nucleic acids. So on the screen, you can see a list. I kind of broke it down of how many you should have left of each. At a minimum right now, you should sort out which ones are your 3 steroids and which ones are your 3 fatty acids. This entire activity will be finished up by the end of the day, Thursday. So today in Google Classroom, you'll see the post for both lipids and nucleic acids. You can finish this up when you have a chance. But pull out your steroids and your fatty acids so you can take a look at those as we continue through today's lecture. So today we're starting to talk about lipids. Lipids are a really diverse group of molecules. There are lots of different structures and functions that they have, but the one thing that they all have in common is that they are all hydrophobic. This is the only group that doesn't include true polymers. There's not one small repeating building block, like proteins are always made of amino acids, carbohydrates are always made of monosaccharides. Lipids do not have one monomer that repeats over and over like that. Many of them are not big enough to be actually considered macromolecules. So that also makes them a little different than the other types of molecules we talk about in this unit. And you can see from the molecules that you sorted out of your set that they are predominantly made just of carbon and hydrogen. There is some oxygen in lipids as well, but they're mostly hydrocarbons. And that is why they're mostly hydrophobic, because hydrogen and carbon form a nonpolar covalent bond. And since they're nonpolar, they don't dissolve well in water. They're very varied in form and function. We kind of already said that. So today we're going to look at one type of lipids. We're going to talk a little bit today about the fats. Fats are made of a glycerol and 3 fatty acid tails. There's only one shown on the picture on the screen, just so that it's a little clearer, but it would have 3 of those fatty acid tails here. These are also known as triglycerides. Tri means 3, so that's that 3 fatty acid tails. The top part, sometimes it's called the head of the molecule, is a glycerol molecule. That's a type of alcohol. It has 3 hydroxyl groups that you can see here attached onto each carbon. The fatty acids are long carbon chains. You can see down the middle, it's this just very long chain of carbon. But on one end, it has a carboxyl group. Here you see that C double bonded to an O with an OH up here. And so the way that these molecules are put together is through a dehydration reaction, just like all of our other molecules. If we remove 2 hydrogens and an oxygen from here, there's our water that we pull out. That forms a bond between this carbon, this oxygen, and this carbon. That type of bond in a fat molecule, in a lipid, is known as an ester linkage. So again, it's still a dehydration reaction, but for every different type of macromolecule, the bond that's formed gets a different name because it has different atoms that are being attached within that bond. So in a lipid molecule, that bond formed through that dehydration reaction is called an ester linkage. The main function of lipids overall, and particularly fats, is to store energy. Fats are really for long-term energy storage. If your body has carbohydrates it doesn't use, it converts them into fats, which can be stored for weeks or months until you might need that stored energy. You've heard probably of saturated fats and unsaturated fats. A saturated fatty acid. So here on the screen, you just see the tail portions of that molecule, because that's really the part that determines whether it's saturated or not. A saturated fatty acid has no double bonds within that central carbon chain. Another way to think of that, you might know that the word saturated means it's like full. It has as many as it can possibly take. A saturated fatty acid has as many hydrogens attached to that carbon chain as it can possibly take. Because this entire fatty acid tail is comprised of single bonds all the way down, it's very flexible, and these molecules can pack really, really closely together. They can just line right up. And because of that, because those molecules pack closely together, saturated fats tend to be solid at room temperature. So think things like butter or Crisco cooking shortening, or lard, things like that, that are solid fats. Even something like chapstick, the fats in there are probably saturated fats because it's a solid at room temperature. Usually saturated fats come from animals, which again, butter comes from an animal product. Lard usually comes from animals. An unsaturated fatty acid has one or more double bonds in the carbon chain. So you can see unsaturated here. This now does not have hydrogens attached down here to these two carbons because those bonds have been removed to kind of form this double bond. So unsaturated means it does not have every single carbon along that chain attached to the most possible hydrogens. Usually these unsaturated fatty acids are in a cis formation, which if you remember when we talked about cis trans isomers, the way you think about that is if you imagine the double bond kind of as a straight line through there, you have two hydrogens that are attached to those two carbons, but in this case, the rest of the molecule is this carbon chain, and those carbon chains are both attached on the same side of this double bond line that we drew. So that's that cis configuration. Cis means they're on the same side. And because of that, you can see that fatty acid chain kind of has this kinked, curved shape. Because of that shape, these molecules can't pack as closely together. And so these are lipids that are usually liquid at room temperature. So think about things like olive oil, avocado oil, corn oil, any kind of vegetable oil. And most of these usually come from plants or fish. Fish oils are also liquid at room temperature and tend to have a lot of unsaturated fatty acids. You might have also heard of something called hydrogenated vegetable oil. Sometimes companies want to take unsaturated fats like this and synthetically convert them to saturated fats by adding hydrogen. But a side effect of that sometimes is that they end up making some fats just as a result of this process that instead of being in the cis configuration, have that trans configuration. So again, if we draw our line straight through the double bond, if that's kind of the plane we're looking at, now our two carbon chains are on opposite sides instead of being on the same side. So up here, this is the cis configuration. The carbons are on the same side of the double bond. The trans configuration has those two carbon chains on opposite sides of that double bond. Trans fats have been linked to coronary heart disease, so you've probably heard about that before. So these are produced as a side effect of this chemical process, and they're not necessarily good for our health. So if you've heard of trans fats, that's what that means, is these unsaturated fats that are in the trans configuration. Again, shape of molecules matters a lot, and it can have health effects in this configuration in ways that it may not when it's in the cis configuration. We are going to continue on Thursday talking about a couple more groups of lipids and also very briefly touching on nucleic acids to wrap up these organic molecules. If you have any questions, feel free to shoot me an email, and I will talk to you tomorrow.